Hello everyone, this is Mr. Jackson, and today we're going to be doing Unit 3, Lesson 3, where we're discussing how we regulate the cell cycle. You'll know you've mastered this lesson when you're able to explain how the cell cycle is regulated or controlled, and you should also be able to compare how cancer cells are different from other cells. So some key questions that we've discussed so far that we're going to be building off of. Why do body cells divide? Well, the primary reasons are so that we can grow and so that we can repair injured or damaged tissues. So that's to say that cells can't just keep getting bigger and bigger. They must remain small. This helps us maintain a high surface area to volume ratio. And that's just another way of saying we want to have as much outer boundary, as much cell membrane as we can to serve the amount of volume that's within the cell. This allows the things the cell need to come in, as well as allowing the waste products that we create to move out so that things don't get cluttered up and so that we can keep things running smoothly. How often do body cells divide? Well, every cell is going to divide at a different rate based on that specific cell's needs. So in the human body, for example, we have certain types of cells that once we create them, they don't continue dividing. Muscle cells and nerve cells are examples of these. They don't continue dividing once they've been developed. And this could be consequential if we were to damage our muscle tissue or damage nerve cells, because we'd have a hard time replacing them. And so these losses might even be permanent. Other cell types, such as skin and bone marrow, the cells in our digestive system, these are always growing and dividing a lot more rapidly and regularly. And I wanted to bridge this conversation from growth and repair to regulation. So cells at the edge of an injury, for example, are going to be specifically stimulated to start dividing and to divide rapidly. So this is because if you were to use an example here like this fractured bone, right, we're going to be communicating that we need to remove these old or damaged cells at the site of this injury, and we need to replace them with new healthy cells, right? So we have to divide the new or the existing healthy cells to create the new ones. And so this is an example where we would stimulate these cells that are surrounding at the edge, surrounding that injury. And we're going to continue laying these new bone cells down until we consider this injury as healed. And at that point, we will actually communicate that information again, and we will slow that process of cell division back to baseline because we no longer need that rapid division. This is just one really common example for why we need to regulate the cell cycle, how quickly or slowly it's occurring. So next question, how do body cells know when it is time to divide? How do we actually regulate this process? So the cell cycle is controlled by what we call regulatory proteins, both inside and outside the cell. And so you can think of these proteins as just ways of taking in information and then communicating that to the cell and then controlling how quickly or slowly the cell cycle is going to continue. So we have internal regulators. These are basically going to respond to events inside the cell. Good examples of these types of proteins, we call them cyclins. And these proteins ultimately act as checkpoints. They allow the cell cycle to proceed only once certain criteria has been met, right? So only once a certain criteria is met do they allow the cell cycle to continue. If that criteria isn't met, they'll either slow the process down or maybe even stop it altogether. And then we have our external regulators. External meaning they're going to respond to stuff happening outside the cell. Great example of this is growth factors. So these growth factors, for example, would allow the cell to speed up, slow down, or altogether just stop its cell cycle. This can be especially important for wound healing, like we've already discussed, right? So the growth factors and these external regulators sense normal healthy cells and damaged cells in the surrounding environment, and that will communicate whether or not we need to speed up or slow down. This also prevents excess cell growth and can keep body tissues from dividing and growing so much that they actually get in the way of each other. And so before we move forward, I want to make sure that we kind of address some of these key internal regulators. We said it's like a checkpoint system, but I'd like for you to also start to think of it kind of like a red light, green light system. It can also be yellow light for slow, but it's an easy way to think about it. So for our cell cycle, we're going to start with interphase, as we have in previous discussions, right? We'll start in the gap 1 phase. So cell begins growing and growing and growing, and then at a certain point, we will get to our first checkpoint, right? An internal regulatory protein is going to check to make sure, do we have enough nutrients? Are there enough growth factors? Is there any damage to our DNA before we continue this process? If there is some sort of criteria that is not met at this point in the cycle, then what we're going to see is that we need to either hit pause, so we could go into G0, which is just a resting state where we're not gonna continue and move forward into S phase until we do have all the nutrients, that we do have all the growth we need. Or we might just stop altogether. If there's so much going wrong that we need to just cancel the ongoing division of this cell, then we'll do that, right? We'll just completely start this process of programmed cell death. We'll get to that here in just a second. And we're not going to move forward with a cell that's not going to be dividing correctly. We'll then go into S phase here at the bottom, where we're now going to be actually making copies of our DNA. So we'll go through DNA replication. Naturally, at the end of this phase, we're going to check, hey, did any damage occur? Did we have any replication errors? Did we copy something wrong? We don't want to make a cell with damaged DNA or with incorrect information, so we would stop it, correct it, and if we can't correct it, no new cell. We get to the G2 checkpoint. Now, this is right before we're going to start division, and we're going to finish that growth. So we're going to, again, check the cell size. Are we large enough? And are there any DNA errors, any replication errors that we can pick up on now? And then finally, we have a M checkpoint during the actual process of mitosis. And this is going to be really important because it's going to check to make sure that spindle fibers actually attach to their chromosomes correctly. We want to make sure that we pull exactly 100% of the information into both new cells. And so if we don't attach correctly, we do not want to continue that process until we do. And so this is a really good example of how these regulatory proteins work and how we regulate the cell cycle and the speed at which it goes down. And I mentioned in the previous slide apoptosis, right? So what happens if we no longer want a cell or a group of cells to continue dividing? We have this term, apoptosis. Apoptosis, you can think of it easy way as just, this is when a cell goes pop, right? So this is when we're going to experience programmed or controlled on schedule cell death. So this is where basically the cell is going to undergo a series of controlled steps. It's going to ultimately self-destruct. So internal and external signals activate certain genes within these cells, and they release self-destructive enzymes. The role of these enzymes is just simply to start dissolving the materials within the cell so that we can reuse them in other cells or in the surrounding tissues. This has a very important role in structuring tissues during growth and development. Can also prevent damaged cells from reproducing. We do not want that to happen. So if we were to, for example, get to one of our checkpoints and we see, hey, something has gone wrong, we can then initiate the process of apoptosis so that that cell dies and we don't continue to reproduce and copy a damaged cell. A very good example of when this occurs is in fetal development, right? So whenever we are in utero, we have webbed fingers and toes, at least initially. The process of apoptosis is how, after we develop our digits, those fingers and toes, it's how we actually program the death of those cells that make up the webbing between your fingers and toes. If that process is interrupted during development, then you might have something like this image down here at the right, where that webbing actually doesn't fully go away. And so you kind of see a little bit of a joining between these two toes for this person. That's just because apoptosis did not continue all the way down to where it normally would. And so that's Apoptosis is programmed cell death. I kind of addressed there what happens if we don't have that normal process of apoptosis. But there's another abnormal part to this discussion, and a very, very prevalent one, which is cancer, referring to what happens when cells divide and divide in an uncontrolled manner. So the essence of cancer is just we have these cells that don't respond to these normal regulatory signals. They just kind of go rogue. They do their own thing. This is when the cell cycle becomes disrupted and it causes cells to grow and divide uncontrollably. Now, we can see the result of what happens when this occurs, right? This abnormal cell division and this rapid cell division ultimately creates what we would call a tumor. And we want to explore the process of that in a little more detail. So you can see here the images sort of representing this process over to the left, and we can describe what's going on. So first, a cell is going to begin to divide abnormally. You can see this cell does not resemble the normal cells surrounding it. And so it's going to divide abnormally. We know at this point that control over the cell cycle has broken down. Cancer is going to ultimately result from a defect in genes that would control this cell growth and division. So something has happened to our actual DNA that has caused complete breakdown of this regulation process. In the second image, you can see here that now cells are going to actually produce these clusters, and we call those clusters tumors. So they start to actually displace normal cells and tissues. They're getting in the way and they're pulling all of the nutrients from those tissues. We do have two types of tumors. We have one type that is benign. These tend to be localized, and this would be a really good example image of a tumor that is benign. Typically, what this means is that it's in a single area, and if it's identified early enough, we can typically just remove it with surgery, right? We can cut it away from the existing tissue. But we do have malignancy or malignant types of tumors as well, which is more represented by this bottom image. Malignant tumors are ones that are able to spread and invade many types of tissues. So in this third part here, if this tumor were to continue developing down to this where we have a blood vessel in this area, they can actually, these cancer cells can actually move into the blood vessel and can move to other parts of the body. And we call this process metastasis, where we actually see a spreading of some disease from one tissue or organ to another. This is where cancer typically becomes a lot more serious because it's a lot harder to treat because it's no longer localized. It's not in just one spot in one type of organ. This could be, an example would be maybe having breast cancer that then gets into your lymph nodes and in your blood supply, and it might metastasize, causing different types of cancer, like breast cancer becoming lung cancer, or liver cancer, and so forth, right? So basically, once it gets into the bloodstream is going to go throughout the entire body, so there's really no limit for how you might develop this cancer further once it metastasizes. And then to finish up our conversation here, we want to close out with what are some of the common causes of cancer. We do have biological factors, right? So like as we get older, there are types of inherited genetic mutations that make us more susceptible to cancer. Skin type, right? So if you have low melanin content in your skin, you're going to naturally be a little bit more susceptible to different types of skin cancers. You have lifestyle choices. So these are the preventable ones, right? Like, so our diet, physical activity, how much sun exposure we get. Typically, good rule of thumb is for diet related that you want to stay away from those inside aisles of your grocery store. If it does not spoil quickly, meaning that you keep it in a pantry and it doesn't go bad, then these foods tend to have a lot of extra processing and extra chemicals involved. So not the best things to be consuming for our body. Also stay physically active, keep your body moving, and wear your sunscreen if you need to. You can also have certain viruses that cause different types of cancers and exposure to carcinogens. So these are just basically chemicals that can actually damage or mutate your DNA. And these mutations, if they happen to be in areas that regulate your cell cycle, that is where cancer starts to develop. So let's go back, break it down. Let's be direct with these learning objectives. Can you explain how the cell cycle is regulated? And I've provided a description of what this process actually looks like. Remember, internal and external regulatory proteins. These cause cells to slow down, can even stop the cell cycle. Something has gone wrong and we don't want that cell to continue dividing, when we can go through what's called apoptosis. And all of these processes are ultimately what shape our tissues and organs. And then, can we compare cancer cells with others? Right? Cancer is uncontrolled growth and division. They don't respond to the normal signals. So this can result in tumors, and these invade the surrounding tissue. Rapidly growing cancer cells can be targeted by different types of radiation therapy. We also have chemotherapy, which is a chemical means of specifically targeting rapidly dividing cells. And we can also surgically remove tumors. Key thing here with rapidly dividing cells and chemotherapy is that this is oftentimes why individuals will lose their hair during chemotherapy treatment, because it's going to indiscriminately target rapidly dividing cells, such as your skin cells, your nails, your hair. So that's one reason why we typically see that. So at this point, we have discussed what the cell cycle is, the different parts of the cell cycle, and now in lesson 3 we have finished off with the specific ways in which that process is regulated. And I hope that you found this conversation helpful. See you at the next one.